Hi. Entner doudoroff pathway is found in many gram-negative cyanobacteria, archaebacteria, and extremophiles, and also in some eukaryotes. But this is considered ancient reaction compared to glycolysis. This actually is also less efficient than glycolysis. As you can see, the products of this pathway is 2-pyruvate, 1 ATP, 1 NADPH, and 1 NADH, whereas in glycolysis you would produce 2-pyruvate, net total of 2 ATP and 2 NADH. It's really not this that are different, but it's actually 1 less ATP that's the difference here. Because together they would also produce 2.5 ATP per each. And NADPH and NADH are pretty much, they produce about same energy amount. And another difference is that Entner doudoroff pathway would produce KDPG intermediate, and this is called 2-keto-3-deoxy-6-phosphogluconate. And if you don't have KDPG aldolase, which is an enzyme that converts KDPG into pyruvate and glyceraldehyde 3-phosphate, it's very toxic for the cell because it inhibits cell growth. The uniqueness about this pathway is that it would use the beginning of oxidative phase of pentose phosphate pathway, and it would go into the midpoint of glycolysis and ends at glycolysis pathway. And glycolysis is also known as Epstein-Meyerhof-Parnas pathway. So if we go in detail for the process, I don't draw out the structures. These are the important names and enzymes that are used here. So this is up to here is the oxidative phase of pentose phosphate pathway. So we would first activate glucose using ATP and hexokinase. Then this becomes glucose 6-phosphate. Then using glucose 6-phosphate dehydrogenase, we are going to produce 1 NADPH. Then it becomes gluconolactone 6-phosphate, which using lactonase and water, it becomes gluconate 6-phosphate. And here, the difference is this enzyme. Instead of gluconate 6-phosphate dehydrogenase, we have gluconate 6-phosphate dehydratase, which releases water to produce KDPG. And here, with KDPG aldolase, we're converting KDPG into pyruvate, one of our products, but there is another part left over, glyceraldehyde 3-phosphate, that's produced. Then this enters into glycolysis pathway, sort of midpoint here. And glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate using G3P dehydrogenase. And here we are going to add this phosphate group to glyceraldehyde 3-phosphate, making it two phosphate groups in this molecule. But at the same time, we are releasing 1 NADH, another final product here. And then 1,3-bisphosphoglycerate is now converted into 3-phosphoglycerate by adding this phosphate group here onto ADP to produce ATP using phosphoglycerate kinase. Then 3-phosphoglycerate is converted to 2-phosphoglycerate. All we did was just moving phosphate group on third carbon to the second carbon, making it more higher energy using phosphoglycerate mutase. Then 2-phosphoglycerate is converted into enol product, phosphoenol pyruvate, using enolase by releasing this water. And finally, phosphoenol pyruvate, very high energy molecule, is converted into pyruvate and producing another ATP using pyruvate kinase. But as you can see, we've used 1 ATP, so our net total is 1 ATP that we produce because they counterbalance. And now we have 1 NADPH and 1 NADH here. And that's the Entner doudoroff pathway.